seminar. It gives me great pleasure to introduce Brendan Carson. Brendan got his PhD from Tufts University in September 2024, where he was co-advised by me and Colin Oriens. And we recruited him to work with us with the plan that he would link sort of chemical ecology and population ecology. And that's totally not what he did. He got really excited in the summer before he came about the potential that a lot of what we were attributing to the environment was actually about a virus. And so he trained himself to do virology of wild butterfly plant interactions. He managed to pull off experiments that involved importing viruses from Canada in 2021. He was actually going to do it in 2020 and that flubbed, but he managed to come up with a completely different experiment to do that summer and has really created his own place in the world. When I suggested he come out and practice here, I thought it might be like a practice for future job talks, but he beat me to the punch, and he'll be starting as a professor at Messina College, which is I don't know what the word is, a little sister of Boston College that works especially to provide a gateway for underprivileged students to college. And I just think that's just the coolest job. So what else? He's from Michigan. He did, I think you're from Michigan. Anyway, he did a master's degree at Michigan State and has also done a lot of work on wetlands and plant ecology, as well as his more recent interest in the diversity we don't see in the world. So, Brendan, thanks for coming.
Thanks, Elizabeth, and thanks everyone for showing up in this talk today. As she said, I'm going to be talking about cryptic viruses and butterfly populations. And just so you know what I mean when I say cryptic, which will come up a few times, there are some pathogens we know are in a pretty high abundance and are really common out there in the world. But there's this whole underlying world that we really don't appreciate and don't know about, and we don't have a very good idea of what its effects on populations and kind of cascading up the food chain really are. And so that's what I mean when I say cryptic.
We're not sure it works as a clicker.
It works as a light, but I think you might need
Okay,
it.
well, I was clicking where it was here.
You just need to click on your presentation.
Yeah. All right. Okay. So I'm really fascinated by the diversity of life on our planet. And for me, a lot of this, especially as, I guess, more as an adult, has come through the lens of looking at plant and insect interactions, partly because these are just such abundant organisms and we see them everywhere we look. You just have to step outside and they're right outside your door. But I'm also interested in all the other parts of life's diversity. So birds, mammals, even people. And but there's this whole world that we don't see. So this microbial world that is also extremely diverse. And we're really starting to understand a little bit more about its ubiquity and diversity through more recent advances in molecular methods. And as we're applying to those to the field of ecology and evolution, really starting to get a better idea of some of the role that these play in our world. So a lot of this work has been really characterizing what's there. So this is a slug, and this is the bacteria microbiome of this slug's gut. So we can see it's very diverse and has a lot of different orders and families of bacteria in it. But names really get us only so far. So it doesn't really tell us very much about what these bacteria are doing. Maybe they just live there and they actually play very little role in that gut's metabolism. Maybe they're really crucial for it, but just knowing those names doesn't actually tell us that. So we know fungi, just to go through a few other microorganisms that are important. You know, fungi both can be mutualistic. They can provide nutrients and water to plants through these mutualisms, through these mycorrhizal networks. But a lot of fungi also are pathogens that can cause disease, either like the chytrid or the cordyceps here. Viruses, similarly, we often think of as causing disease. So this is the barley yellow dwarf virus. We know that this virus and other similar viruses have actually played a really large role in structuring the grass communities in California. So the viruses are actually do better and replicate better in exotic grasses. but those grasses are not impacted as strongly as the native grasses here. There's been a lot of work showing that without these viruses, native grasses would actually be competitively superior to a lot of the exotic grasses here. So again, if we want to understand the diversity and kind of structure of life around us, we really need to think about how the microbial world has the potential to influence it. But we can't just look at the names of those microbes. We really need to study specific interactions and start to understand how they scale up to population level effects. In recent years, there's been a lot of interest in how disease influences both humans and nature. And epidemiological theory has taught us a certain amount about disease dynamics. So one of the most important models that's come from epidemiology are these susceptible, infected, recovered models, SIR models. And they're really the basis of our understanding of how disease spreads through populations. Here's an example of this. This is a time series. So time 0, you have this population of just circles. They're all susceptible individuals. And if one of them becomes infected, now we have this one infected individual, and it can pass that pathogen, that infection, on to additional members of the population. And over time, the initial infection, that individual becomes recovered. And this one that it passed it on, this died. This one spread its disease to another individual. And now over time, we're missing this one that passed away. This one is now recovered, and this one passed it on. So these models are compartmental models that really help us understand how one infection can spread to either one or multiple new infections, and once an individual is recovered, it may be less susceptible to being reinfected. And I think that maybe this was abstract before 5 years ago, and a lot of us learned a lot more about this stuff during the pandemic. So this basic pathogen reproductive rate, or R naught, is one thing that comes out of the SIR model. But then also we get these concepts like herd immunity, the difference between an epidemic and an endemic disease, density dependence, and a lot of other phenomena that are attributed to disease ecology come from just that basic SIR model. So one of the assumptions of that model is this idea that transmission increases as population density increases. And so here we have a population increasing. And then if there's negative density dependence, then we might see a decrease as that population reaches a peak. And then you could actually have some cycling happening as a result of that. So in order to see a pattern like that, you need to both have density-dependent transmission, but then also the impact of disease must be strong enough to cause population decline. So there's a lot of diseases that we think of as being pretty severe that don't actually cause strong population decline. This is especially true in human populations. So in the past, there have been all of these major epidemics. You can see our population, maybe if you look really carefully, you could see some dips at the time of these pandemics, but overall our population has continued to increase. So these are examples of diseases that can cause strong mortality, but don't necessarily impact overall population dynamics. So again, if we want to understand the effect of microbes on diversity in life, we really need to understand the role microbes play in shaping species distribution through the lenses of natural history, microbiology, population ecology, and epidemiology. So really need to combine a lot of different fields in order to really understand how a given disease or pathogen or microbe has the potential to impact populations. So I think this is exciting because we're at a moment in history where a lot of the molecular tools are a lot more accessible and less expensive, and people are talking across disciplines a lot more than I think that they used to. So I'm going to present my dissertation research as a case study for how this might be done. The study system I've been working in is the Baltimore checker spot butterfly and the densovirus that's called the Johannesini densovirus. That's the caterpillar of the checker spot there. And the driving questions here are, is the impact of disease strong enough to cause population decline? So kind of like looking at that graph of human population over time, those really weren't causing strong declines, even though they were severe pathogens. And then also is transmission density dependent. So we're going to start to investigate some aspects of this as well in this talk. So here's Colin Oriens. This was one of my PhD advisors, and then Elizabeth Crone. They're on the right. And as Elizabeth said, I joined Tufts really with the idea of working with both of them because there were parts of both of their work that I was really attracted to. And Elizabeth had been studying a population of Baltimore checker spots, actually many populations, but one in particular, we had seen this exponential increase, so this big boom followed by a strong decline. And I showed up in 2018 with my butterfly net, all ready to go out and collect some butterflies, and lo and behold, there were not a whole lot left at that point, at least there. And I started really wondering, well, what could have caused this decline? And we knew that there were still plenty of host plants available for them to lay eggs on. And we knew that there was not strong evidence of parasitoid wasps at this population that can have a big impact in this system, but there wasn't great evidence that that was what had happened here. We also had this observation that the female to male sex ratio was declining at the same time that the population was decreasing, which was an interesting observation that we didn't have a good explanation for. So I started thinking about then the possibility of pathogens to cause this population-dependent decline. And the first thing I thought of was monarch butterflies and some of the work that's been done in a parasite called OE. I can never remember its actual Latin name. It's very long. It has a lot of Z's and other consonants. But anyways, I started wondering, like, maybe there's a parasite like that could be impacting checker spots. And I had a friend that had just finished her dissertation work with Mark Hunter at University of Michigan, who had studied OE in monarchs. So I called Leslie Decker up and asked if she had any idea whether there were other parasites like that in other butterflies. And she did not know about that, but she actually texted me a photo that she had taken at an ESA conference of really recent work of a virus that had been found in Baltimore checker spot butterflies in New England. So, so it was a lab based out of University of Nevada, run by Angela Smolanich. This is a figure from their first paper on this. And it's basically, you don't need to focus too much on the numbers. It's just showing that each population they surveyed, they were finding pretty high or relatively high abundances of this novel virus, this Johannesini virus, in populations across New England. So of course I started wondering, well, maybe this is responsible for that decline that we saw at the checker spot population. So I started learning more about this virus, the Johannesini virus. And really, if you look at an electron microscope image, they're pretty nondescript. It's really just these little circles. And I thought this was kind of a funny statement. Electron microscope images of native JCD-MV virus particles reveal a homogeneous population of small spheres with few evident surface features. So it doesn't really tell us much about what it's doing. So we know that this virus is in the family of viruses, parvoviruses, that are the same things that cause dog and cat parvovirus, but densoviruses are all infect invertebrates. So it's this group within that all evolved to have invertebrate hosts. And there's single-stranded DNA viruses, invertebrate hosts. There's a lot of diversity, and it's largely undescribed. So we think, you know, if every insect had a densovirus that affected it, it's probably not true, but it's possible that it could be that much. You know, that's a huge diversity just within insects. And there's a lot of other invertebrates out there too. There's also evidence that densoviruses can cause population decline. So this is an example from sea stars. Here's one from these beetles that are farmed for pet food. But there's also some examples of densoviruses actually having a positive impact on their hosts. So it's possible that some of them are actually mutualistic, while others are parasitic or detrimental. So just because we found the presence of densoviruses in checker spot butterflies doesn't actually show that they're having an impact at the population level or a negative impact. So first, I'm going to focus on this. First question is the impact of disease strong enough to cause population decline? Or is it even disease? I guess we should also step back and ask that. And is transmission density dependent is the next thing I'll be talking about here. So our roadmap, can a cryptic virus drive negative population growth in a charismatic butterfly species? Second, I'm going to focus on movement across heterogeneous habitats and ask how can habitat heterogeneity influence disease. Really, this is the one that's drilling into density-dependent transmission. And then finally, I'm going to briefly talk about a side project that was focused on community metagenomics. All right, so I've already introduced this butterfly a little bit. This is a great model system. There's been a lot of work done on checker spots generally, and that includes ones out here on the West Coast, like the Edith's checker spot, but also on the East Coast and central US, the Baltimore checker spot. There's been a lot known about it and a lot of studies done. So one thing that I find intriguing is that it could actually have this cryptic thing that is actually an important factor in its populations that was just flying under the radar this whole time. So something really interesting about this species, and actually the original reason I got interested in studying it was that it evolved on a wetland plant called turtlehead, but it actually, a lot of populations now use a non-native, this is a European plant, English plantain, or lance leaf plantain. And they both have these chemicals called iridoid glycosides. And it's really this chemical similarity that's allowed for this host plant jump. They also have meta populations. So across the landscape, you have populations that are discrete, and you can study them as a single entity. You can go out and capture them and mark their wings. So you can do mark recapture and figure out how many individuals are present and what their survival is and things like that. And you can, because of this metapopulation nature, you can observe new populations emerging and old populations declining and disappearing. And you can ask, well, why did that population decline? So this brings us back to this population. I started to think, well, maybe this virus was playing a big role here. Okay, so we can't actually go back and study this population decline after it happened. So what I settled on was an experiment that is really investigating the impact of virus across each life stage of the butterfly and then using that to put together a demographic model. And it's possible that it could be a negative impact or a positive impact, or we could see differing impacts in different direction actually at different life stages too. So like many insect species, checker spots have very high reproductive capacity, and this could possibly buffer them from pathogen-driven decline. So you think about humans, like a lot of humans have died from these epidemics, but we have pretty high reproductive rates, maybe not as individuals, but I guess as a total population, we're able to mitigate that. And maybe in insects, that often happens too. So is the impact disease strong enough to cause population decline? We need to understand the impact across the entire life cycle. We want to understand its effect on population growth. So here's the life cycle for this species. I'll start, I guess, with the adults. They're active for only 2 or 3 weeks in the summer. They lay their eggs on one of those host plants, and those eggs hatch, and the little tiny caterpillars, they lay their eggs in big batches is another key thing here. So hundreds of caterpillars can all be siblings and form these gregarious webs, and they'll overwinter together as 4th instars. So after they've gone through a few molts, they'll overwinter under the leaf litter and emerge in the spring. And it's really in the spring when they start feeding really vigorously and doing a lot of growing. So they reach this kind of stasis period and then overwinter, and then they emerge, and at that point they're feeding a lot, they're intaking a lot of plant matter, and their metabolisms are really ramped up, and they start moving around and foraging, which could expose them to more virus. We'll talk about that part in a little bit. And then they pupate, go through metamorphosis, go back to the adult stage here. So we really want to figure out at every one of these life stages, what is the potential impact of the virus? So an instar is a word I'll be using. It's really just the caterpillar life stage. And then diapause is just essentially like hibernation in insects. So this first field experiment I did in 2 years, 2021 and 2022. And something interesting about those years is that in New England, the 2021, we had a lot of rainfall, and in 2022, it was a pretty severe drought year. So we got to look at kind of a climatic effect that we hadn't planned on. So what I did was I sprayed a solution of these viral particles on plants, host plants that I planted on campus in little cages that were all in an enclosure. And I used control, had no viral particles added. And then essentially as you went up, it was an increasing dose. And this is actually a pretty big jump here, because we had some evidence that there's kind of this range at the low end where there's no impact. So we wanted to really look at the biological relevant range of virus here. So this is the experimental setup, sprayed it on, and then introduced a known number of caterpillars to each cage and followed their survival. So first, looking at egg to second instar survival, we had a known number of eggs, and then I went back and counted how many made it to the second instar. And I only did this in 1 year. And so I'm going to show you a series of figures. On these figures, the x-axis is the virus load that they were exposed to, and then the y-axis is the survival, or there's a couple other metrics we'll look at too. There's not really an impact seen here for egg to early instar survival. And then I looked at pre-diapause survival, so from the second instar all the way to when they overwinter. And for this, I moved these little tiny caterpillars onto, I had to count them into batches of 30 and then move those onto the experimental plants. And we actually saw very little or no impact of virus in either direction at this stage. And then we looked at overwintering. So I knew how many of that pre-diapause stage had survived up until the end of the fall. And then I went back periodically in the spring and counted how many actually emerged in each cage. Oops. And here we actually saw a little bit of a positive impact. It's marginally significant. And I thought that was pretty interesting, because I really actually expected this to probably be the strongest negative impact, because overwintering is just a really stressful time for insects. Okay, and then we get to post-diapause. And so this is essentially putting in, I kind of restarted the experiment at this point, because I assumed a lot of the viral particles had washed away during the winter and early spring. And so I resprayed a known quantity of viral particles on the plants and introduced 8 of these early spring caterpillars, and then followed them through pupation and counted how many adult butterflies emerged in each cage. And here we saw a really dramatic decrease in both years of survival as virus increased here. Okay, so here in this life stage, we see negative impact. Then I looked at the sex ratio, because it's really the females that are contributing to the next generation. So this is important for a population model. And you can tell the sex by looking at this little patch where And so this was interesting. So in year one, we actually saw no impact on the sex ratio. In the second year, we saw pretty strong negative impacts. So females were less likely to survive in the second year. And then finally, looking at fecundity, the number of eggs each female laid. I let the females mate with males from the same treatment if they survive, assuming they survived, and then put them in oviposition cages with their preferred host plant to lay eggs on, and then counted how many eggs each female laid. And here we saw a significant negative impact in each year of the exposure to virus. All right, so then we have all of our life stages put together. So then we need to combine the vital rates for each life stage. And so we can look at it all like this. We see some go up, some go down. It's pretty hard to tell what's going on here. So we use this basic model with lambda, and then survival rates for each life stage multiplied by the sex ratio multiplied by the fecundity. And so here, lambda is a population biology seminar, so I think you probably all know this, but if lambda is at 1, the population is stable. If it's above 1, we have an increasing population, and below 1, it's a decreasing population. And so I use some bootstrapping to generate confidence intervals in these estimates. But what we saw was that for the first year, which was the wetter year, we saw a decline in population, a decline in lambda as virus exposure increased, but it actually never got below 1, even out at the very high virus exposure levels. And in the second year, so the dry year, we see overall just a lower growth rate in general. And then also as virus exposure increased, we crossed that threshold pretty early on. And by the time we get out to the highest rates, we're basically at, it's not quite to 0, but it's very close. So it's to the point where there's just almost no survival. And so this is that data from the site that Elizabeth had been following that showed the increase followed by the decline. So I just talked about precipitation here, but something to note is that this decline took place during the period of drought. And then we also see that in that wild population, we saw this decrease in the female to male sex ratio around that same time. So, you know, it's not nail in the coffin evidence by any means, but it is an interesting correlation that in the wild population, we saw a few similar things that we saw in the experiment I did. So is the impact of disease strong enough to cause population decline? Yes, sometimes climate seems to be an important factor here, and sex-specific mortality really exacerbates the impact. And the strongest effect is on this post-diapause life stage. And this is interesting, I think, because this is really the stage where they're going around and eating the most, so they're going to be exposed to the most virus in the wild, assuming that the virus is on their host plants. And they're really mobile. So this is a stop motion video I took. And you can see they're coming and going. They're eating this plant, and they're also depositing pieces of frass caterpillar poop on it when they're doing that. So I'm not going to really talk about this experiment, but one thing I did was I showed that if you inoculate caterpillars with the virus, you can find quite a bit of it coming out in their frass, and actually just as much comes out in their frass as we found in their bodies. And so if you think about caterpillars crawling around, feeding and defecating, then that's potentially going to spread virus around their habitat quite a bit. Okay, so this brings us to our next question. Is transmission density dependent? For this, we looked at movement across heterogeneous habitats and asked how can habitat heterogeneity influence disease? So again, thinking about this SIR model that all of these principles are based on, this model is really based on, or relies on this assumption that as population density increases, the transmission rate is also increasing. And one thing that it really assumes is this linear relationship here. So we know transmission is proportional to host contact rates. So really this is coming from this idea that as population density increases, individuals in a population are making more contact with each other. So we can switch transmission for contact here and really think about it in these terms. This really results from another assumption, and that's that every host in a population can interact with every other host in the population. And this is known as homogeneous mixing. However, non-even use of habitat can create spatial structuring that disrupts this assumption. I think we all can probably agree anyways that that's not a very realistic assumption in the first place, that every individual is going to equally interact with every other individual. So I really started thinking about how the structure of habitat and kind of where females were deciding to lay their eggs was impacting contact within caterpillars in these populations. So one thing that really helped us in this is that the checker spots lay their eggs in these big batches of 50 to several hundred eggs. And so they form these webs that are helping protect them. We think these are to help them protect them from parasitic wasps, but they're actually easy to find. So rather than needing to go out and look under leaves and find every single caterpillar, you can go out in the late summer, early fall, and look for these. And because they're all in these aggregations, you can actually survey every member of the population and have a pretty good idea that you've captured all of them if you use mercury capture methods. So if we think about the distribution of caterpillars, we think about host plants kind of being evenly distributed across the site, which isn't always the case, but just for this, we're going to think that. So really where the caterpillars end up is a function of where the females decide to lay their eggs. So the females maybe are going to be attracted to areas that have a lot of flowering plants, but maybe they're going to be attracted to moister soil areas because this was what is a wetland species. So in either case, they lay their egg, and that's where the next generation of caterpillars starts their lives. So we assume that there's this uniform distribution. That's what where our linear relationship is really coming from. But in reality, we might have a random distribution, or we might have a clustered distribution, especially if females all have similar preferences in where they actually choose to lay their eggs. And contact results from organismal movement. So we have large movements of these caterpillars as they're foraging in that spring generation or that spring time period. In this case, we think about caterpillars moving around versus that scenario. You actually achieve homogeneous mixing because any caterpillar from any part of the site can actually encounter other caterpillars that started anywhere. And in this case, this linear relationship is actually maintained. However, if you have short movements, then individuals are really going to only encounter individuals that started nearby them. And you don't have homogeneous mixing. However, in this random distribution, you actually maintain this linear relationship. However, if you have clustered or kind of patchy distribution across the site, then we don't know what the shape of that relationship looks like, and it actually depends on the specific orientation of where caterpillars are. So I wanted to really investigate this empirically, and so what I did was I looked at spatial data for nest locations and combined it with models of caterpillar movement to estimate contact in real populations. And here we ask, is transmission density dependent? So we're focusing on, one is distribution of nests, random, clustered, or uniform. And then if the distribution is clustered, does this moderate density dependence? What is the scale of movement relative to the scale of heterogeneity we might see? And then finally, does resource availability influence the amount of movement we're seeing here? So I used larval spatial data, nest data from 6 sites in 16 site years. And these were data that Elizabeth let me use that had really been collected by past Chrome Lab members. And each site has a different population density. So this was kind of one of the key things in this study, was that by using a lot of different populations that each represented a different, or kind of had data collected in a different year, we're able to get a range of population densities to look at these relationships. So we have, this is an example of 2 different sites in a given year. These are observed locations of where we found nests. And then I also simulated random locations using the same population density. So across both of these are both same density, same site geometry, but either the observed distribution or a random distribution, a Poisson distribution. And then I used movement tracking to come up with a movement model. So I had 2 sites, one with abundant host plants and one with host plant scarcity, where I followed caterpillars. And I did this across caterpillar development, so through these 3 spring instars, to think about, well, first of all, as they're getting bigger, we expect they're probably to move a little bit more. And then also their total movement is cumulative through time too. Here's an example of a movement path. I put a caterpillar down, or I followed a caterpillar, and every minute I put in a pin where it was, and then took a photograph of its movement path and then traced it out. And so from the lengths of each step and then the turning angles, we were able to get some basic statistics about movement here. And from this, we were able to come up with diffusion coefficients to figure out how far we would expect them to move over their lifetimes. So here in this example, we have these 2 plants, and female butterflies laid eggs on each plant. Each of these is starting out a given distance away, so let's just call it 20 meters, just for fun. And so these aggregates of larvae are overwintering together. And in the spring, they emerge. They pretty quickly eat that host plant that they were laid on. And then they start to disperse into the surrounding landscape. So I use these 3-dimensional distributions. So it's essentially just a normal distribution in 3 dimensions. And each of these, it represents where each of those plants was. And so this is after the first instar. This is how much dispersal we expected to have happened on average. And over the second, this is how much. And so we can see here that they're starting to kind of bleed into each other. And by after that last instar, they've dispersed even further. And so essentially all I did was quantify the amount of overlap in these distributions, and then did that for every pairwise distance between each set of nests here. And so I did that both for our observed distribution and then for these random distributions that I generated, and then looked at how much contact we expected to happen in each scenario. So first of all, did we actually see clustering? Like when I was going out in the field, I thought I was seeing clustering, but we wanted to actually quantify this. And so here is this clustering metric. And if things were random, we'd see this falling out here. If things were uniform, which you can imagine if females actually wanted to give their larvae as much resources as possible, maybe they would try and space them out across the landscape. If they were clustering, then maybe they'd go that way. And so all of our caterpillars were way over here. So there really is a lot of clustering happening within these habitats. And then combining this with movement data, we have, just the movement data by itself. So the diffusion coefficient you can kind of think of as a proxy for how far they're moving. And I looked at this again in an area with low host plant availability, the light gray, and high host plant availability. And so you can see in each instar, when there's less resources, the caterpillars are moving more. So that, I think that makes sense if you really think about it. And that's what past literature has really showed for a lot of other animals is that they tend to move more if they need to find more food. Talked about that already. Okay, so if we put these things together, then we can estimate the contact that's happening across these different sites. And again, the different sites really represent different population densities. So here is a kind of a big figure, but on the x-axis, we have population density for each site. And then we have up here, this is assuming slow dispersal. So this is if there's a lot of plant resources, food resources. Here we have fast dispersal. So if there are not as many resources available. And another aspect of these figures is that up here, you can see that the black lines are the observed distributions. The gray lines are going to be the random distributions. And then finally, these panels are 4th, 5th, and 6th instar. So how much contact we expect to have happened as time progresses during development. Okay, so just looking, if we assume that there's slow dispersal, we can see in the random, if we're looking at the random distributions, the simulated distributions, in each case, there's this linear relationship, just like the assumption of the SIR model, and it increases as population density is increasing. If we look at our observed distributions, for this first 4th instar, so when not very much movement has happened yet, there's actually no relationship here. So even in the most dense populations, we actually don't see a lot of contact happening. In some of these less dense populations, they actually see more contact. And this is really happening because in some of these sites, the females are laying their eggs really close together. And so even though there's a lot of habitat available, we have a lot of contact occurring at these lower site level densities. And then as development happens, we do see this positive relationship, but it never is this linear relationship. So it's kind of asymptotic. All right, so if we assume fast dispersal, then we do see a positive relationship here. It's asymptotic. And then as development increases, then we actually shift into this expected distribution that is, or it's a linear relationship. And so essentially what this is showing, if we look at just these, is really showing that the movement is really an important factor in determining the shape of the relationship between contact and population density. So and then we're also just seeing more contact as a whole and when there's more clustering. So if we had random distributions, there's just less contact happening. So the fact that these female butterflies are choosing similar locations to lay their eggs is just ramping up the amount of contact that's happening within a site. So is transmission density dependent? Yes, but it really depends on site-specific resource geography. So we can't really just always assume this linear relationship, and we might be able to assume it's generally a positive relationship, but its shape is really site-specific. So implications for this are that context really matters when we want to think about disease spread within a site. And to a theoretical ecologist like Elizabeth, this might be a little bit depressing because you can't just plug your numbers in and come up with the real answer. But to a naturalist like me, I think this is really neat actually, because it's showing how much kind of the diversity and geology and human use history in any location on our planet actually has a big impact on not just the diversity of organisms there, but actually those interactions that can cascade up to population level impact. All right, I probably don't have a lot of time left, but I'm gonna just briefly talk about this last project. It's Lepidoptera and community metagenomics, what other viruses we find in co-occurring moths and butterflies. So when I was doing this, I really started thinking about the idea that in a population of checker spot caterpillars that is really big, then if there's a disease here, it has the potential to spill over onto all of the other moth and butterfly species that are present. And also, I didn't put this figure, but we could have the reciprocals. So all of the other individuals could be contributing diseases the other way too. So most studies of eukaryotic viruses are really focused on one host and one virus at a time. So that's really how we think about most of these epidemiological models is one at a time. So for instance, here's a little brown bat with a virus that affects it. So most of these models would go like this, but really there's multiple viruses that affect that bat. And there's a lot of other mammals that have other viruses and some of them may or may not be shared. So if we think about these caterpillars in a field, there's a lot of other caterpillars that are living with them there, and they may or may not be sharing viruses between them. So this was really kind of the impetus for investigating this. And so what I did was I went to 3 different sites that had Baltimore checker spot populations, and I just spent a lot of time walking around with a sweep net and collecting caterpillars. And then I would use some forceps to pick them out and then put them into a container. And I would do as much field sorting as I could, but then I would bring them back and sort them under a dissecting scope and put them into groups of all the same species. And so I found almost 3,000 caterpillars, 30 species with more than 8 individuals, and a lot that had fewer than that. And we do 28 individuals per sample. And so then what I would do, I'm not going to go into the details, but essentially I would surface sterilize them because really we were interested in what was inside them, not on the outside. And that was to try and minimize cross-contamination as much as possible. And then I had a protocol that was geared towards first really trying to enrich for viral particles by filtering out bigger stuff, and then extracting DNA and RNA, and then amplifying that and sequencing that. So I used with Illumina sequencing. And then I used some bioinformatic tools that allowed me to, unfortunately at this point, only get down to the family level of viral diversity. But even looking at that, we can't necessarily answer the ecological question I set out to answer, but it's a really kind of neat lens into how much viral diversity there are in these systems. So this is an example of one site in 1 year. So these are all different caterpillar species here. And then these are all different virus families. So if it's filled in then with a purpley color or a white color, then we found that virus family and that caterpillar species. And these are all insect viruses out to here. And then I also found a lot of plant viruses, which makes sense because they're eating plants. I also actually found some bat and bird viruses, which I'm guessing just came from feces on the plants. And then a lot of bacteriophages, which are viruses, bacteria. And so some of these were really, we found across most of the species of caterpillars, and others were more rare. And so this is each site in the 2 years. You can see there's just a lot of viral diversity there. So I did some other sort of work with this that I'm not going to share because I think having to stop at the family level for identification prevented me from getting into a lot of the cool ecology that could result from this if I'm able to tease this out in the future. But I think the thing that really this highlights to me is that there's a lot of cryptic interactions happening in these systems that we just really don't know about at all. Like we don't appreciate at all how much viral diversity and fungal diversity and bacterial diversity there is, and they have the potential to be impacting populations. A lot of them probably aren't, but I think a lot of them probably do have some impact too. Right, and I think that's up there. All right. We quickly acknowledge a lot of the undergrad students that helped with this work, and also Elizabeth and Colin were really pivotal. And also, this is Angela Smilanich at University of Reno, Nevada, and I learned a lot from her about working in this densovirus system. And then Lee Brown was a postdoc with Elizabeth for a long time, and she really helped train me in the checker spot aspect of the system too. So shout out to them.
So it's 5 o'clock, so if anyone needs to go, I will say aloud that you are allowed to leave. But otherwise, we have time for questions. Oh, look at me, look at him.
I don't want to intrude on other people's time. I have a whole train of questions, which depend on your answers to each one.
Okay.
Stop at any time, and if I become obnoxious, please, either you or Elizabeth tell me to shut the hell up.
All right, sounds good.
For those who don't know who I am, I'm Art Shapiro. I work on butterflies, and I have a history going back to the 1960s with this bug. Okay. You had some feeding on turtlehead and on English plantain. English plantain, as you said, is a naturalized weed from the old world. It was actually one of the very first exotic weeds documented in New England by John Jocelyn in the 1700s. Okay? But as far as I know, the Baltimore checker spot was never recorded on it before about 1960. Am I correct on that?
Yeah, I think maybe even 70s. Yeah.
Well, it was on it in the
Yeah, yeah.
60s because it was already on it when I was a graduate student at Cornell in the 60s.
Oh, okay.
Okay. You described your densovirus as a Junonia Buckeye virus. Was it originally described from the Buckeye?
Yeah.
Okay, this is becoming very interesting.
Yeah.
Because English plantain is a major host plant of the Buckeye.
Yeah.
But of course, they're not co-evolved because the weed is introduced. The Buckeye is an extremely mobile, even migratory species. Okay. Which raises the possibility that your virus got into the Baltimore checker spot as a result of it feeding on English plantain, which was contaminated by Buckeyes.
Right, so it's the result of using this novel host plant.
Right.
Yeah.
Okay, that's a hypothesis. Okay. So that leads in a number of interesting directions. You have 2 other checker spots in New England, Harris's and the Silbury checker spot. You're in a different genus, Closini, but they're closely related. Do you happen to know whether the virus has been found in either of them?
No, I don't. Yes, I don't think I didn't find them in any of my surveys, and I never went out specifically to.
Okay, you probably know is with Harris's checker
I
spot.
do know there's a checker spot. Although, before we have a long conversation, I'm gonna ask if anybody, especially grad students, have any
Yes,
questions.
please, I don't want to monopolize.
But we do want to talk to you more. You know, Peter's practically a grad student relative to Art.
Yeah. I'm applying. I'm applying. All right, so I loved your sort of one of your conclusion slides from the piece of work that you did that this would depress Elizabeth, but it made me happy. I thought that was a sort of a cool result. And I think what I wanted to ask about that is, have you thought about kind of what the meta implications of those 2 possible pathways are? And did you sort of come up with ways in which that's influencing the system, or would have influenced it very differently if it in fact had conformed?
So I think I know what you're getting at, or sort of, and so the main way that I've thought about it is that, you know, I think there's only so much we can do about disease impacting species of conservation concern. I think that the basic approach I could think of is just factoring in the effect of disease if you're making a plan that assumes some kind of average population growth to a given species. And I think what these results suggest to me is that even in smaller sites, smaller population, well, I think I need to step back a little bit. So you could, I think if you looked only at the theory, then you would assume that as population size decreased below a certain threshold, then the disease would stop, it would die out because there wouldn't be enough contact between infected individuals for the disease to persist. That's kind of the idea that when you fall below the population density that allows on average one transmission of an infection to a new host, then the disease falls out. But I think what this suggests to me is that because even in a small population, if the individuals of any species all have similar preference in their habitat, then they're probably going to be congregated a lot more than you would expect than if you're just kind of assuming these. And I think that's probably obvious to most ecologists or animal watchers in general. But I think it was interesting to me to really go through the steps to show it. Yeah.
I was curious when you were doing the calculation for the population growth rate stuff, when you were going through like each like life history stage and saying like whether or not like you saw a change in things like survival and such. I was curious when you were doing the population growth rate calculations, were you only considering ones that like did come out as like significant in the statistics?
Oh, that's a great question.
Or did you just do
So I just used the effect size, even if it was not significant.
Gotcha.
Yeah, and I did think about which way to go, and that's kind of what we ended up on. Yeah.
Well, did you try the other way? Like, was it like fairly robust if you only did?
I actually don't think I ever did the whole analysis the other way. That would be interesting to do that though. See, yeah.
Thank you.
Yeah.
I know nothing about virus ecology, so I'll just preface my question
Yeah.
with that. Do you have any sense of like, what are the seasonal dynamics for the virus itself and how that might be influencing both prevalence and effect at the different life stages?
Yeah, that's a good question. So, I mean, this is really like a like winter, spring, like it's a very seasonal system and I think there's a lot of the natural history of it that's not really known, but my guess, so it does seem to be pretty or what would the right word be? It's like it's robust. So like COVID is only, if it's on a surface, is only going to persist and be active for 3 days or so at the most. This, if it's not exposed to ultraviolet light, like let's say it's underneath a shadow in some leaves or in the soil, it seems like it could persist for years actually. That being said, I think there's like this decay rate over time where if you have a big infection or outbreak in a population, then they're going to be depositing a lot of new viral particles into the environment. And then you're going to have more of them are going to be present the next year. So they could potentially have a bigger impact that second year. But then once the population, like let's say it actually does cause the population to die out of hosts, then you would expect over a lot of time for them to decay, unless they're able to use alternate hosts, which is kind of where I started thinking about that whole community dynamic thing. Because if that's the case, then the environment becomes less, kind of that environmental stability becomes a little bit less important if they're also present in a lot of the other caterpillar species that are present.
So now it's 10 after, so I'm gonna let people go, but if you have more questions, then you can come up and chat with Brendan and perhaps Art and I afterwards. Thanks, Brendan.